Good morning, ladies and gentlemen. It's a really nice pleasure to meet you here. Especially, this is the first time I've been in Lithuania. My name is Patryk Matuszek. It's written like that. And I come from Poland. I've been working for the Euroimmun company for the last 5 years. This is the company maybe you know. That still are working on new methods for diagnostic of autoimmune diseases, infectious as well as allergies. And because today we are talking about tick-borne diseases, this is why I'm going to discuss two main things. The first thing is modern diagnostic of Lyme disease. I'm going to talk about some criteria, international criteria. I will show how it's done in Poland and in such countries like in the United States. But let's start with some basic things. We are talking a lot about Borrelia. We think we know a lot about that, but we have to know this is still young disease. The first time we saw the problem in last century, in 1975, it was in the United States in the city that was called Lyme. This is why we are talking about Lyme disease. And 10 years later, a man who was named Burgdorfer isolated the spirochete and called it Borrelia burgdorferi. In the United States, we have only one human genus species, which is human pathogenic, but in Europe we have at least 4. So the same like in the United States, and Borrelia garini, Afzeli, and Spielmani. In the down right corner here, we can see the smear of spirochetes, which are transmitted by ticks. The picture here shows the development cycle of tick, which starts with small larvae through the nymph and the adult tick, which is very important for us because we have to remember that tick on every stage of its development it's human transmitted. So sometimes we see the problem with a patient who have symptoms, have antibodies, and they say no, it's not possible because I've never been bitten by ticks. So some people don't remember, especially if they are bitten with something small like this. In Poland, we first saw the problem of Borrelia in 1996. In that year, we reported about 700 cases of Borrelia, and as you can see, the amount of reported cases was increasing through the years, reaching even 10,000 cases in 2009. So I don't think it's the problem is not we are infected more and more, but as we know, we know the problem better, we have better diagnostic methods, so I think it's just reported cases. At the picture here we can see the tick during the dinner, so in human skin, and here we can see the tick after lunch, so fits with the human blood. Through the years, we were trying to develop many different laboratory methods, which can be divided into groups, which we call direct methods, like microscopic tests, culturing, as well as PCR, and indirect methods that do not detect Borrelia itself, but something else, for example, antibodies. I have to say that direct methods are not recommended in many countries. Why? Because the big disadvantage of these methods is that positive result is only possible to be received only when in the tested material bacteria is present. So you can imagine that, for example, I'm infected with Borrelia right now, but for example, I have it in my knee, but the laboratory draw my blood and test it with PCR, so I will I will receive so-called false negative result. This is why we recommend to use indirect methods that detect antibodies in our blood. So no matter where the bacteria is situated, the antibodies are detectable in our blood. So we use such methods like ELISA test, which is a screening test, and confirmation test, which is Western blot test. The first country that released some official recommendation, it was United States in 1995. 5 years later, also Germany released such recommendation. And in Poland, we also have the same recommendation, which was released by Polish Association of Epidemiologists and Infectious Disease Specialists. If you read these three recommendations, you will see that they begin with two common sentences. The first sentence is that the base of diagnosis is clinical image. So we have to have symptoms. We don't treat the laboratory result, we treat patient. And the second common sentence of these three recommendations is that there is only one specific symptom of Lyme disease, which is called erythema migrans, as you know. It's so specific that we don't need laboratory tests. If we have another symptoms than erythema migrans, then we need to have laboratory tests, which is recommended in 2 steps. In Poland, as a screening test, we treat ELISA test that detect IgM and IgG antibodies. Every negative result is the final result, but every positive and borderline should be confirmed with Western blot test. In some countries like United States, also immunofluorescence is recommended as a screening test. Here we can see the rule of immunofluorescence. As an antigen, we use smear of bacteria on a microscope slide. Then you incubate it with human serum, and if there are some antibodies present, they bound to the antigen. In the second step, we incubate with conjugate, which is labeled with fluorescein. And the example of such test, as you can see on the one field, which is for one patient, you can see 2 different genus species of Borrelia burgdorferi and Afzeli, and 2 main antigen, OSPC and VLSD. I'm going to describe them later. There are 2 ways that you can release result from immunofluorescence. It can be qualitative result, simply positive or negative or borderline, but it's recommended to use semi-qualitative results doing dilution series of human serum. So you can see example here. The picture is not too good, but you have to believe me that the titer of antibodies here is 1 to 1000, because this is the last dilution. We still observe positive signal. We have to remember that we use different starting dilution for IgM and IgG. For IgM we start with 1 to 10, and for IgG we start from 1 to 100. In Poland, we recommend to use only ELISA test as a screening test. The rule is very similar to immunofluorescence. The exception is that conjugate is not labeled with fluorescein but with some enzyme, and you can see the product of enzymatic reaction as a colorful product. Ladies and gentlemen, there is a big problem in Borrelia diagnostic because on the one hand we have recommendations that say please use ELISA as a screening test, but on the second hand there is nothing mentioned about antigens. So depending on the company that produce ELISA test, they can produce according to its own ideas. Some companies produce ELISA based on full extract of antibodies, some select the most important, and some of them produce recombined antigens. I just want to show you how we created our ELISA. So we can see the Borrelia here. Of course, it's not Borrelia, but just a picture that have many different antigens in its cell wall. We take all of these antigens, so we produce the full extract because we can produce antibodies against every of these antigens. But you can see the other bacteria that also have some specific antigens for in its cell wall, against which we can produce some specific antibodies, but you have to see that Borrelia and this other bacteria have the same antigens. There are some similar in some way, so we can produce some unspecific antibodies. So these blue antibodies give us no information if we are infected with Borrelia or with another bacteria. Screening test should be the most sensitive it can be. This is why we do not select antigens. We take the full extract of antigens from Borrelia. We coat ELISA well with them, and then you incubate it with human serum. If the patient have had a contact with Borrelia in the past, the antibodies are bound here. Of course, we receive positive result. But we have to remember that every of us is different. So here's an example of patient that produced every antibodies he could, but maybe another patient that also have contact with Borrelia produce these two antibodies only. But of course, the full extract of antigens detects this patient as well as the third example of patient that also have had a contact in the past, but produce only this one antibody. But of course, we will detect him. So the big advantage of using full extract of antigens that we will probably find every patient that have had a contact in the past. But of course, such system have also one disadvantage. So maybe in the future you will meet the patient that have never had a contact with Borrelia, but maybe with some other bacteria, and produce these unspecific antibodies. Of course, in that case, we will receive so-called false positive result. This is why every positive result which is received on ELISA test should be confirmed in Western blot test. Why? Because Western blot test should answer the question if the antibodies that cause the positive reaction in ELISA, these are the specific for Borrelia, or maybe some unspecific produced because of something else. Here you can see the example of our ELISA test. So these are these wells coated with full extract of antigens, and these are full extract of antigens from Borrelia burgdorferi, Afzeli, and Garini, and also one recombined antigen VLSC. I will talk about it later. Our ELISA tests are also validated for testing CSF, but every laboratory that wants to diagnose neuroborreliosis, because it's for neuroborreliosis, should be sure that have two more tools in the laboratory. Because our ELISA test, it's just for detection specific antibodies against Borrelia in serum and CSF, but we also have to calculate albumins in these two liquids and total IgG or IgM, because we want to calculate so-called CSF coefficient. Why? Because we don't want to detect only antibodies in CSF, but we want to answer the question if they were produced in central nervous system or maybe diffused by blood-brain barrier. There are no standards, there are no international standards, so it means we have no international units. So depending on the company that produce ELISA tests, we have different units. Here you can see relative units that validate our ELISA test. So over 22 relative units, we treat the result as a positive, behind 16 as a negative, in the range 16-22, it's borderline result. So ladies and gentlemen, we have done ELISA test, we received positive result. Now we want to answer the question if this positive result was caused by the specific antibodies or maybe unspecific. In recommendation, there are mentioned Western blot tests that evaluated through the years. So now I don't know any laboratory that use this classical Western blot. Most people in Poland use combination of Western blot with line blot technique. I will show you later. And the newest things is the confirmation test based on recombinant antigens. We can talk about Western blot only when electrophoresis is done. So this is the process of production of Western blot test. We take the same antigens, full extract of antigens from Borrelia afzeli in this case, put it on the gel and separate it in the electrical field. So now the same antigens that are coated in ELISA test, now we have every antigen as a single band. In the second step, we do electroblotting, so we transfer antigens from gel to the membrane. The membrane is cut on the single strip, and the single strip is confirmation test for one patient. You can see the example of such positive result. So we can see separated antigens. Some of them are specific, some of them are unspecific antigens. Example of two different patients, the patient behind. This is the patient with fresh infections. So because of the symptoms erythema migrans, I told you that if we have erythema migrans, we don't need laboratory diagnostic, but we tested this patient to show maybe there are some antibodies. And of course, in IgM, we detected antibodies against OSPC. And antibodies against OSPC that in most cases are the first antibodies that appear in our blood. So for many physicians, so for many diagnostics, this is the marker of fresh infection. The patient below with late stage of Lyme disease, we see no OSPC antibodies in IgG, but we see many different antibodies against many different other antigens. So we see that immune system of this patient has evaluated. The evaluation of this test can be done manually, but we created special software which is called Euroimmun Scan. You can scan the strips, and the program will analyze every positive band on the strip, like in this example or in this example. This is quite interesting example. We took some series from CDC, from Center of Disease Control from Atlanta, of the patient with fresh infection. And what can be observed, that most of the patient with fresh infection had antibodies in IgM against OSPC. So this is the evidence that these antibodies may be the marker of fresh infection. But you can also see this 4 patients here, that on the one hand are patients with fresh infection. They are infected, but we see no positive result. So this is the false negative result. Why? Because probably they are tested too early. So the second problem we meet in Poland are with the patient that say, yesterday I was bitten by tick and I want to be tested. So we have to remember that serological window lasts about 4 to 6 weeks. So we have to tell to the patient, okay, but wait at least 4 weeks and come back, then I will test you. Here we see the results of healthy people. But what can be seen that although they are healthy, there are some antigens present on the strip, but there are unspecific antibodies. So probably they obtained the positive result in ELISA, but now we can see, for example, the flagellin. This is the protein which is present on many different bacteria against which we can produce antibodies, but except of flagellin, we see no specific for Borrelia. So this is the example of negative result. Some people in Poland ask, maybe we can finish with ELISA. Maybe if we have positive ELISA, maybe we can treat the patient. Of course, no. So you can see the evidence here. For example, this 27, 11, and 8 patients, so in total 46, that obtained positive result in ELISA. It appeared that not every of them is positive. Some of them are borderline, but 8 of them is negative. So for example, if we finish with ELISA and take the antibiotics, it's not necessary. So please all the time do confirmation tests when ELISA is positive. I showed you the first very important antigen, OSPC. We talk about OSPC, that antibodies against OSPC in IgM may be the marker of fresh infection. But now I want to show you the second very important antigen, which is VLSE. Here you can see the short history of discovery. So in 1986, a very interesting protein was isolated on non-pathogenic Borrelia hermsii, which was called VMP, from English variable major proteins. Some years later, this very similar protein was discovered on Borrelia burgdorferi, so we call it VMP, VMP-like sequence, so the same sequence like VMP, so it's called VLS. And what appeared? That on the one hand, it was a very specific antigen for Borrelia. On the second hand, it was very immunogenic proteins. So antibodies were produced against VLSE, and these antibodies was very, very specific. This is why we wanted to take this antigen as a specific antigen for our test. But for many years, bacteria was smarter than us. We saw no expression of this antigen in vitro. This is why we have to do recombined antigen, which is called VLSE. And here you can see the structure of this antigen. This yellow-red color. This is the variable site of this antigen that still change and cheat our immune system. This is why, in contrast to OSPC, antibodies against VLSE appears later. So this is marker of late-stage infection. So what we did, we took this Western blot test that we saw in some slides before, and put one additional membrane with recombined VLSE, which is situated at the end of this strip. And what is this the big advantage of such solution? The high sensitivity. We increased the sensitivity about 12% in contrast to classical Western blot, and now this test is much more easy to read. Why? Because if we do the test in IgG and you see the VLSE positive, this is positive result. You don't have to take care of this rest antigens. The same situation is in IgM. If you see the positive OSPC, you release positive result in IgM. The prevalence of the antibodies against VLSE is also very interesting. The patient with erythema migrans, about 30% of this patient have antibodies against VLSE. But for example, patient with neuroborreliosis, even every patient have antibodies against VLSE. What does it mean? That this is more marker of late infection than the fresh infection, in contrast to OSPC. But the sensitivity of this antigen is about 86%. What it means that in 86% of every positive result, you see the positive VLSE. So in 86% of positive result, there is very clear diagnostic. You don't have to take care about these rest antigens. This is the correlation of classical Western blot and Western blot together with VLSE. Especially interesting are these 15 people that was undetectable by classical Western blot. Now we can say they are positive because they have antibodies against VLSE. So it means that the sensitivity increased about 12% in contrast to classic Western blot. This is the example of result which is released by Euroimmun software that I showed you previously. What can be seen of the result? The scan of the strip. I told you that not every antigen is specific. This is why we analyze only the specific antigens for Borrelia, and the information for our patient, if it's positive result or negative or maybe borderline. Ladies and gentlemen, when I go to some conferences like this one, I meet two groups of people. There are people who say that native antigen is better, so the antigen I showed you. But there are some people who say that no, recombined antigen is better. Who is right? I think these two groups of people are right because every solution have advantages and disadvantages, right? If you are talking about native antigens, we have to say that such solution show higher sensitivity because we use every antigen from Borrelia. So we will detect more positive patient, especially if we add VLSE antigen. But of course, there are some disadvantages of such solution. In more times, you will meet this borderline result. We don't like borderline result, right? Because of the presence of such antigens like P30 or P31, and such antigens like P30, P31 that differs only with a 1 kilodalton can be situated on the same place in the electrophoresis, so sometimes it's very difficult to distinguish. If we are talking about recombined antigen, we have to say that we can apply antigens from many different genus-species. In classical Western blot, it's not, we cannot do it. But here we can put antigens from Borrelia afzeli, from Borrelia burgdorferi, Garini and Spielmani, for example. The production is simple and the evaluation is simple as well. But disadvantages of recombined antigens is lower sensitivity. Why? Because we have less antigens and there are no recommendations which antigens shouldn't be applied. So every company that products such confirmation tests can use different antigens. So one patient tested in 2 different laboratories may receive 2 different results. So this is big disadvantage of such solution. But we have to say that recombined antigens passed the exam in diagnostic, for example, of antinuclear antibodies, antibodies in autoimmune liver disease, in neuroline antigens. So we hope that recombined antigens will help us in diagnostic of Borrelia as well. How we created the test? We analyzed the genome of Borrelia, selected some immunogenic antigens, did 2-step selection and selected only this specific. So now I'm going to show you the example of such test. As you can see, we have 3 different VLSC from Borrelia afzeli, burgdorferi and Garini. The new thing is lipid antigens, which are marker of late infection, and some recombined antigen as well. Through the years, it was believed that the best source of OSPC is the native antigen. Why? Because the OSPC in the membrane of the bacteria is the dimer protein. If we product recombined antigen, we receive monomeric form. But in the last year, our engineers managed to produce recombined OSPC antigen, which is very, very similar to native one. This is the test for IgM. So we can see OSPC from 3 different genus species, from Borrelia afzeli, burgdorferi, and Garini. And the newest test, which is called Advance, that apply this newest OSPC antigen, which is called Advance from Borrelia afzeli, burgdorferi, Garini, and Spielmani as well. So here we can see the positive result of such test with the recombined antigens for IgG and IgM. The new thing is 2 additional control bands for conjugate. So if you are doing the measurement in IgM, you should see the IgM positive band. If you are doing the measurement in IgG, you should see the positive IgG band. So summarizing, it's very important to do 2 steps laboratory diagnostic. So in some countries, immunofluorescent may be a screening test. In Poland, we are talking about ELISA only, but every negative result of ELISA or immunofluorescence test is the final result, but every positive or borderline should be confirmed with a Western blot test or the Euroline with recombined antigens. So here we can see the example of immunofluorescence test or ELISA, which are screening tests. The classical Western blot with VLSC, so this is the one recombined antigens, and the alternative to classical Western blot is Euroline test. Thank you for your attention. If you have